chapter, we're going to be talking about vertebrates. But I say essentially, okay? So the phylum, okay, it's not vertebrates, okay? It's chordates. Now, vertebrates are part of the chordates, okay? So this tells you that there are some chordates that in fact are invertebrates, okay? The very first group. We're going to talk about who they are, okay? So not all chordates are vertebrates, they're most of them. Now, before we zoom into the chordates and look at the characteristics, why do we classify a chordate as a chordate? There must be some features there that make us do this classification. And we'll look at these main features that you would like to know very well from me and what they are. Before zooming into that, I wanted to give you an overview of what we are going to study. So we started from something like a flatworm, right? We said that a flatworm, remember, breathes through the skin. Okay, remember, these are very sensitive to pollution because they breathe through their skin. Okay, then we moved on to insects, which do develop some kind of respiratory system, some kind of complex, if you will. But it's not very efficient, right? This is the tracheal system that we just looked at, remember? Series of tubular structures that become ramified inside of the body of the animal. And as we will see, the respiratory system becomes much more specialized in fish through the formation of gills. And finally, we have evolution of the lungs. Okay? And we're going to understand these gas exchanges, how they are really developed very finely, very efficiently through evolution. Okay, they become very, very efficient. Oxygen and CO2 exchange machines. This first the gills and then the lungs. Okay, we're going to go over these. I also have some nice videos for you. All right, so phylum Chordata, chordates. What's chorda, cuerda? Remember when we did talk a little bit about this when we were doing the classification, I think it was a lion. Who remembers? What did I tell you? But the lion, at some point, we saw that belongs to the phylum Chordata. If you know a little bit of Spanish, cuerda is a rope, okay? So Chordata have all these rope-like structures, but not necessarily as adults. The features that we look at are embryonic features. You know, we do this sometimes. We do this when we look at embryonic development to establish all these milestone stages of animal embryos, remember, that we looked at. They make us actually classify an organism as an animal. An organism as weird, as different as a sponge, goes through the same embryonic milestone stages as us, okay? Besides the few cell stage, then there is the morula, the blastula, and the gastrula. And that's one of the strong points that makes us classify a sponge as an animal, okay? As weird as that is. Now, in this case, also, we look at embryos, okay? Things may change in the adult, it doesn't matter. If the features are present in the embryo, that's what we look at, and that's what makes us classify a chordate as a chordate. So the very first structure that even gives name to the group, to the phylum, Chordata, is this notochord, okay? We call it notochord. It's a rope-like structure that supports the body. Now, could this become a spine in the adult? Yes. If that's the case, then we're talking about a vertebrate. If it stays as a gelatinous material, pretty strong though, that supports the body, then you are a chordate, but you're not a vertebrate, okay? Okay, so this is the first feature that even gives name to the group. There are three other features that all chordates have. Did you know that you have a tail? Where is it?
the end of the spine.
It's been kind of reabsorbed. But if you look at the human embryo early stage, it has a little tail there. This tail is post-anal, okay? Comes posterior to the anus. So all embryos of chordates have a tail, okay? They may not conserve it as adults, but all embryos have a tail. And if you look on the internet, well, be careful because there are some false pictures, but there are some real pictures of children born with a little tail because it has not been reabsorbed. During, errors happens, mistakes happen. Be careful of the artifacts though. So, the other two characteristics are a nerve cord that is hollow and is dorsal to the notochord. It runs dorsally to the notochord. Okay, not ventrally, but dorsally. And then, did you also know that you have gills? Yes, you do. During the very first steps of your embryonic development, you have gill openings. You have openings there in this area. Okay, now those openings will close up, and the structures that were supposed to be structures that support the gills will become part of your early airway system. Okay, but you pretty much go through a stage where you have gills. Okay. So the gills, basically, they're called either slits or pharyngeal slits, or even just openings. You'll find several ways that they are defined. Okay? So this is a schematic representation of an embryo of a chordate. Okay? 4 elements to be known. Okay? That characterize chordates. Tomorrow, if I find a new species, I'm not sure how to classify it. It's very small. It looks maybe like a fish, but it's not really a fish. If it has these 4 characteristics in the embryo, I am going to place it in the box of chordates, right? Because I have established these are the 4 characteristics that distinguish chordates. Okay, so notochord, a rope-like structure that supports the body of the embryo, is a gelatinous structure. It may become calcified, it may become spine. If that's the case, then I am in front of a vertebrate, okay? But in some animals it doesn't happen. So they are chordates, non-vertebrates. And this is the dorsal hollow nerve cord that runs dorsally to the notochord, okay? The pharyngeal slits or openings here, and the post-anal tail, the 4 characteristics of chordate embryos. They make us, again, classify chordates as such. Again, just because I like to emphasize what's important, okay? Chordate features, pharyngeal pouches, also they're called as the pharyngeal slits, or gill openings, you can call them, that's fine. Hollow dorsal nerve cord, hollow and dorsal to the nerve, the notochord, or that rope-like structure, post-anal tail, and that's it. The 4 characteristics of chordates. So you must remember 4 me. All right. This is a human embryo between 4 and 5 weeks. Oh, look how cute this tail is. Isn't it cute? Okay. You really do have a very visible tail during embryonic development. I think the pharyngeal slits or pouches are really closed up here, but I see a spine already formed there, so the notochord is already turning into a spine, so this is definitely a vertebrate, right? Of course, we are vertebrates, okay? All right, so again, before going into the groups, the various groups of chordates, let's try to understand what are the trends here. Okay, we start from the most the simplest group, and then we see that there is more complexity that adds on with time. That's a trend in evolution, right? And so what are these transformations that happen? First of all, the notochord at some point becomes becomes mineralized, becomes bony, and so we become from having a gelatinous structure supposed to body to have a bony structure, a vertebrae that support the body, a spine. And these will end up containing the nerve cord. The nerve cord, we'll see that how the very first chordates don't really have a skull, okay? Then we have the appearance of a skull, okay? We'll see who there are, the groups that don't have a skull. Then we have evolution of jaws. We go from jawless to jawed types of fishes, okay? And later on, okay, so we'll see how chordates appear in water. And chordates are the groups from which vertebrates come from, okay? So we'll see how at some point there is a shift from life in water to terrestrial life, okay? So we used to live in water, we were fish, okay? That's who we come from. And this memory is even conserved in our embryonic development, so much so that we have gills during our embryonic development, okay? So initially the aquatic groups are fins, but these fins become limbs. This was a fin. Could it become a fin again? Did it become a fin again in some organisms? A wolf-like creature comes from a fish. A wolf-like creature, by selective pressure, could become a whale, right? Oh, there is so much plasticity in evolution, not good limitations. Okay, yes, of course, can become that again. It all depends on the environment. Obviously, it takes millions and millions of years for that to happen, okay? I know it's hard to think about this kind of timing. It's hard for me, I can imagine for you. When I start thinking about this kind of timing or geological timing, my brain gets lost. I just can't, it just cannot understand what 1 billion of years means. You know, it's a number, right? But we can't really immerse our mind into that, right? For me, it's very hard. And so we go from breathing with gills, because we are in water, and that's a good system to breathe in water, to breathing with lungs, okay? And we'll see how interesting this shift is. Do you know that there is some type of fish that you can drown for keeping it in the water for too long? Yes, there are, okay? We'll see these interesting groups that represent a passage between the life in water to life on land. Okay. So these are the trends in evolution, and we'll talk about them step by step, okay? These are the general trends. All right, so I already told you when we get to this phylum, maybe for each phylum we didn't go much into the detail of the various classes, which is the next classification box, right? We stayed kind of general, but here, because this is the phylum where we belong, right? We belong. We like to zoom in a little bit more and try to understand really what is the step-by-step evolution here. All right, so in this phylogenetic tree of chordates, okay, you have an ancestral chordate, some chordate that evolved this chorda mostly. They gave rise to first group, which is the group of tunicates, which don't even have a brain, okay? And then a second lineage with a brain, but the second lineage didn't really have a skull, okay? Until a real skull, a real head now appears. Okay, so it tells you step by step what are the innovations, okay? Here going down into the phylogenetic tree. Okay, so the first three groups of chordates don't have a vertebral column that evolves, that develops into the adult. So these are the invertebrate chordates. You see that the vertebral column starts appearing here, okay? And it's conserved. But these first three groups don't have a vertebral column that develops in the adult. So they are invertebrate chordates. So remember, not all chordates are vertebrates. All right, tunicates. Tunicates are also known as sea porks. It's very strange little organism. You would never say that this is our ancestor, would you? Okay? They go through a larval stage, okay? They live in marine environment. Again, the adult doesn't even possibly resemble any shape that we have looked at, but if you look at the larva, you will recognize the bilateral symmetry and you recognize all the features that I told you of chordates. You will recognize the pharyngeal slits, you will recognize the notochord, highlighted in green, you will recognize the hollow nerve cord, and you will recognize the post-anal tail. So the embryonic or the larval stage in this case has all the features of chordates. So this is a chordate, okay? We don't care what's going on with the adult. If at the embryonic stage we see those characteristics that I told you, it's a chordate, as weird as it can seem. Okay, tunicates, probably not so familiar to you. Lancelets are actually most known in Asia, especially because they are eaten, okay? Very much. They are very, very tiny. They look like fish, okay? But they are not fish. And if you zoom into the body, you'll see all the features that I have talked about. Now, the notochord, okay, stays as a gelatinous material. It's not a fish because a fish is a vertebrate, okay? But it does have that shape, and you could very well mistake it for fish, okay? Lancelets, okay, the second group. This is the real picture of how they look like, okay? Okay, all the characteristics. Third group is the hagfishes. You probably have seen this in an aquarium. I don't know, and many of you. Hagfishes have, first of all, let me tell you, I'm going to tell you that they have a cranium. And so cranium is a chamber that initially is cartilage. Is a material that is not really bone. Do we have cartilage in our body? Mm-hmm. Point out to it. Here, so it's a flexible material, okay? And so initially it's cartilage, then become bone. That encloses the brain. So that's the cranium. Okay, you should all know this term if you didn't already. And so hagfish is a craniid, so it does involve a skull. Okay, it's not a vertebrate. Remember, it does not have a backbone. It only has a notochord, the gelatinous notochord. It's a bottom scavenger, produces slime. Never seen one of them. Okay, so these again, they seem like almost like fish. They're not fish. Okay, they're not vertebrates, and they have different characteristics. They have those four characteristics for chordates, but they're different from fishes. Okay, from fish. All right. So these first three groups again, they were not vertebrates. Now there is one group, the group of lampreys. I don't know if you have ever seen those ones. These parasite fish in salt water. Now this group is a little bit in between the invertebrate chordates and the vertebrate chordates. So it does have some characteristics, makes us classify them as vertebrates, but in reality they don't really have the true vertebrae that develop. They have a series of cartilaginous structures, they're called arqualia actually, that are arranged above the notochord. They're the beginning, okay, of a vertebral system. They're the beginning. So we do classify them as vertebrates, but they're not quite that. Okay, you know, there is always a passage. Okay. All right. So lampreys, and that's all I'm gonna tell you about this first group. Okay, not too much. Now you see that And so we started with evolution of a vertebral column already here. So in lampreys and on, you'll have a vertebral column. Now with next group, which is the group of Charcteris, a real vertebral column. And here you also have evolution of jaws. So lampreys and all the rest are jawless. Look at the lamprey, they don't really have a jaw, okay? Now we have evolution of jaws, okay? Where do jaws come from? Do they come out of the blue, or they must be a modification of a pre-existing structure? Check 1 or check 2? Go with check 2, right? And so, where do jaws come from? There's a bone system, okay? And so they come from modification of structures that are around the gills, the very first gill openings that are meant to maintain the gill opening. That door didn't have a frame around or collapse, right? So a gill opening, you need to have a frame around it to be maintained open. Now, that's a structure that gets modified over time, over the course of millions of years, and it becomes the bone system of the jaws. And so the next group that we're going to look at, which is the cartilaginous fish, sharks, rays, are jaw fish. Okay, so before zooming into the cartilaginous fish, let's define what's a fish. A fish is an aquatic vertebrate, 100% vertebrate, that respires, means breathes, using gills, and is ectothermic. If you analyze this word, you should know what that means. Ectothermic. What can it mean? Ecto means outside. Thermal refers to temperature. So, what does that mean?
It gives off.
It keeps the temperature, whatever.
It does not regulate its internal temperature like we do, right? Its internal temperature reflects the outside, the ecto, outside temperature, okay? The temperature of the environment in which it lives, okay? So these animals don't regulate internal temperature, and we'll see when their regulation starts. It doesn't start until much later on, okay, in evolution. Has fins and has a skin that is usually covered with scales, okay? And I have here the definition of ectothermic for you guys to know, this works for you. Endothermic is an organism that regulates its internal temperature and maintains more or less constant. Okay? All right. Fish respires through gills. Gills are a super efficient machine that allows a very efficient and fast exchange of oxygen and carbon dioxide as well. Gills are composed of lamella. They're repetitive structures in which in each lamella there is gas exchange. Now, you know that through a lamella, oxygen is going to be taken from the water into the blood of the animal, or vice versa. Where is the direction of oxygen going? You have a series of structures known as lamellae, okay, which are in the gills. In each lamella, there is gas exchange. Now, oxygen is needed by the animal. So what's the direction of oxygen from the water inside the body, or vice versa? Number one, who says number one? Why just two people? Where does the oxygen need to flow? From the water inside the body. Because the cells need oxygen, right? Why are you super perplexed? It's like, seemed like a tricky question. Was it so easy? I hope. And you know that during metabolism in our body, body of all animals, there is production of CO2, carbon dioxide. Carbon dioxide needs to leave the organism. every time we exhale, we breathe out the carbon dioxide that is formed in the cells of our body through metabolism, okay? So there is not just oxygen going in, there is also carbon dioxide going out, okay? And so I want you to watch this video that explains and it shows you the pictures as it explains and the animation about this process, okay? It's a very sophisticated but also very logical and simple process.
The structure and function of fish gills. For animals that live in water, obtaining oxygen is a particular challenge. Water is more viscous and dense than air, and water contains a much lower percentage of oxygen. This figure shows how a fish's anatomy helps it obtain oxygen from water. Here, the blue arrows show the flow of water into the mouth, over the gill arches, and out. Let's take a closer look at how gas exchange occurs in the gills. The gill filaments are made up of lamellae, and you can see in this closer view that the lamellae are a series of flattened plates. Water flows past the lamellae in this direction, and blood flows through capillaries in the lamellae in the opposite direction. Oxygen-poor blood returning from the body tissues to the gills is shown in blue, and oxygen-rich blood returning from the gills to body tissues is shown in red. Since water and blood are flowing next to each other but in opposite directions, this is called countercurrent flow. Over the course of this countercurrent flow, gases are exchanged between the fluids. We'll focus here on the movement of oxygen. Countercurrent exchange is very efficient. Let's look at why. We're now looking at a single lamella. For simplicity, we're showing just one capillary, but you can still see the progression from oxygen-poor blood on this side to oxygen-rich blood on this side. You can also see that the direction of blood flow is opposite of the direction of water flow.
This is the key to this process, okay? The opposite direction of the flow of water and blood through the lamella. That's the key. Now I was going to show you and talk about oxygen concentration in partial pressure. Don't worry, it just means concentration. Okay, when we talk about a gas, oxygen is a gas, we talk about partial pressure, so don't get thrown off by this word, it's just the concentration that it's talking about, okay? So listen to now what happens here.
The numbers on the arrows represent the partial pressure of oxygen at each location. Will oxygen undergo net diffusion between the water and blood at this box location? And if so, in which direction? Yes, into the blood, yes, into the water, or no net diffusion. A gas always diffuses from where its partial pressure is higher to where it is lower. At this location, the partial pressure of oxygen is higher in the water, where it is 120, than in the blood, where it is 110. So here, oxygen will diffuse from the water into the blood in the capillary. Now let's look at how countercurrent flow enables gas exchange all along the gill. When blood enters the gills, it encounters water that has given up most, but not all, of its oxygen. The blood picks up a little oxygen and moves a bit farther along the capillary. Now the blood has a slightly higher partial pressure of oxygen, but it's also encountering water that has given up a little less of its oxygen. Since the partial pressure of oxygen at this new location is higher in both water and blood, but is again greater in the water than in the bloodstream, oxygen still diffuses from the water to the blood, and the blood picks up more oxygen and moves farther along. This exchange occurs along the whole length of the capillary, with the blood continuously picking up oxygen and encountering water with a higher oxygen concentration. As it reaches the far end, the blood has become oxygen-rich, but it now encounters water that has not yet given up any of its oxygen. This water has just started its journey through the gills, and its partial pressure is higher than anywhere in the bloodstream. So the blood picks up even more oxygen. You can see that oxygen diffuses from the water into the fish's bloodstream along the entire length of the capillary, as shown here by the black arrows. This is what makes countercurrent exchange so efficient, and why the fish gill is an excellent example of the evolutionary relationship between structure and function.
Okay, so to summarize, you have this countercurrent flow, okay? The key is that, the countercurrent flow that ensures that at any given time in the lamella, in the blood that's flowing through the lamella, the concentration of oxygen is lower than in the water. And you know, hopefully you have this concept of diffusion. If I put a drop of food color in a glass of water, you'll see that from its very concentrated where the air drops down, it starts diffusing. So any substance, no matter if it's a liquid or a gas, will always diffuse from where it's more concentrated to where it's less concentrated, okay? That's a concept that you should already have in your mind. And so the countercurrent flow ensures that there is always higher concentration in the water than in the lamella, and therefore there is diffusion of oxygen at any given point through the lamella. Diffusion of oxygen is a very clever, very efficient system of exchange of oxygen, okay? Where it's more difficult in water environment, is oxygen, because of viscosity of the water, compared to the air, is much more difficult to obtain oxygen. But this system ensures a very efficient exchange. Now, the reverse thing happens for CO2. It's the same thing. CO2 is going to be always more concentrated inside the blood that flows through the lamella versus the water, and so it's going to diffuse out. Because remember, we want to get rid of CO2. We don't want to conserve it into our body. Whether you are a fish or a human, it doesn't matter, you want to get rid of the CO2. Okay, so talking about heart circulation in fish and heart structure, heart in fish is very simple, okay? It only has like two chambers, an atrium and a ventricle. We'll see how heart becomes much more complex and a much more efficient machine, if you will. It becomes a perfect machine, actually, in birds and mammals as well. So we start with a two-chamber system, okay? And where there is one direction of circulation. This is not very efficient. You'll see how when the heart becomes really sophisticated, you have actually a double circulation, okay? Here you have one direction where the heart is pumped through the gill capillaries. There is one convention here I should probably mention to you, but most of you may already know. The blue signifies what kind of blood, and the red signifies what kind of blood. Oxygenated. The red is always oxygenated, the blue is deoxygenated, okay? I forgot to mention that, but it's something that most of you know already. Okay, so unoxygenated blood is pumped through the gill capillaries, get oxygenated, and goes to the systemic capillaries, which is the capillaries that serve all the organs in the body except for the respiratory ones. Okay, it gets deoxygenated, it goes back to the heart where it gets pumped again through the gill capillaries. It's a unidirectional circulation. We'll see how, as I told you, it becomes multi-directional or bidirectional. Okay, so heart circulation and heart in fish vary very simple. All right, so jawed fishes, or we just talked non-jawed. Now we talk about jawed fish. These are two classes. There is the cartilaginous fish and the bony fishes. Who are the cartilaginous fishes? Sharks, okay, rays. And from there we become bony fishes. Okay, cartilaginous fishes. Several differences between cartilaginous fishes and bony fishes, not just the composition of the bones. Here we have cartilage, and cartilage is very different from bone. You know, you can flex it, right? It's a flexible material. And I have a tabular representation here where it shows you other differences between the two, okay? But besides that, there are a few more differences, okay, besides the skeleton of cartilage, between the two groups. And this is just a schematic representation of the anatomy of a shark and all the fins, which are pretty much the same fins, the type of fins that we find in fish, with the dorsal, pelvic, anal, caudal, pectoral. There are sharks that can reach very, very huge dimensions. Many sharks actually they don't eat not even other big fish, all they eat is plankton, okay, just like whales. So we have this idea that sharks are dangerous. Yes, there are some that eat big prey, including humans, but they are not all like that. Okay, so as I will talk about bony fishes, I will point out the difference between cartilaginous fish and bony fishes. There are several elements, several characteristics that bony fishes present that a shark does not present. The first question that I have for you, I guess we're going to talk about this difference next time, but I have a question for you before I release you. Have you ever noticed how a shark swims? It goes like this or in circles. How does a fish go? If a fish wants to go up and down very quickly, it goes like this, right? So this is a fundamental difference, and there is an organ that bony fishes have that sharks don't have, okay? It's a sort of a balloon in their body that can fill up very quickly and with gas and compress and release this gas that makes them have that movement very quickly up and down. These organ sharks don't have, okay? So that's why they need to go like that, okay? And in order to maintain their buoyancy, they use a very fatty liver. We're gonna talk about this on Thursday. We're gonna see each other, right?